Okay, so the second part of this lecture on research misconduct, we will discuss two things. One is on biased sampling. Second is, of course, the most obvious, falsifying data, making up data. Okay, these two parts. Now, this is not easy to discuss. Perhaps you don't have any research experience, but at least you should think about the possibilities to make mistakes before you really start a project, right? So even for your final year project or senior research projects, you are asked to write a proposal. So suppose before the proposal, you should, in the summer, you try the experiment out, right? You see whether the test is valid or not. You want to know if there's any measurement problem, right? You want to know whether the method is reproducible or not. So if you use the same machine and then you go to the same site and you assume that the data will be the same, but every time you get different measurement, that could be a problem. And then number 3 is on control. Now that is also not easy to find a relevant control, especially for environmental sampling, because the environmental conditions could be very different. Of course, you will say all samples or all experiments have a control, but most of the time we have more than one control. We have a blank control, we have a positive control, right? You put the drug in, you know that this drug would induce this enzyme. Now I'm going to test a compound whether it would induce an enzyme. Then you need a positive control. And then you also need a negative control. The negative control usually is what you call a vehicle control. Or even more fancy, a control, this chemical would not do this job, so I'm using this chemical. So you have a blank control, vehicle control, negative control, and positive control, and then your own control. So most of the time, if you read biochem experiments, you can see one set of data, and then you have different parameters, right? Plus, minus, there are different combinations. Okay, say like my student would like to study the effects of copper, and then would like to compare with cadmium. Then you have to do copper, you have to do cadmium, and then you have to do copper and cadmium. You may do P-exposure with copper, and then you add the cadmium. And then you do co-exposure of copper and cadmium. Okay, a lot of headache. That is to avoid biased sampling. And fourthly, considering the amount of experiments that you need to do, not enough sampling size. 196 well play, I could only allocate 12 samples, so I have to control the negative control and so on. So, okay, number of samples would be 3. And usually, you know that 3 cannot produce significance. And then, best if the sampling size is 6, and then, I'm doing RNA sequencing. Each sample is so expensive. I could only do 2 samples because each sample is so expensive. I only have $10,000 or $10,000. Okay, I had some good funding available. Then perhaps I could do more samples. But you cannot do this and write this in your paper, right? Because of lack of funding, so I could only give you this. But that could happen. So you have to be very careful how to justify to use enough sampling size. This is particularly true when you work on animal tests. Because one of the basic questions that the Animal Ethics Committee, they are clinicians, they are from the hospital. Usually they publish one paper with one patient as a case or a case study. And then they will ask, could you justify why you have to use that many fish in your experiments? That is the most commonly encountered questions in my research. Oh, I have to use n equal to 5, then I have 5 tanks. Each tank I will put 4 fish, then each treatment I will have 20 fish put in. I have to explain, and then they come back to me. Could you just put 2 fish into each tank? Then I'll say, oh, because the sample weight is not heavy enough for me to do RNA extraction. So I need to, oh, then you have to pull the sample together. So there are many different sampling issues that you have to watch out. Data entry and statistical force. If you use 1 tank, you have 5 fish, your number of samples is 1 or 5. You have 1 tank, you put 5 fish into a tank. So your number of samples is 1 or 5. Not 5, 1. Then you have to do free replicate. For each treatment that I'm doing an experiment on microplastic, testing its effect on jellyfish. So I got 3 tanks. Each tank would put 5 jellyfish. There's jellyfish swimming around. If I have time, I'll show you this video. Very lovely. And then I add microplastic onto it. Right? But 1 tank, a sample number is 1. You cannot say, okay, I have 1 tank, then I put 5 fish, then number of samples is 5. No, that will create a lot of statistical problem. Okay, I do this experiment and number of samples 3. Obviously, it's not good enough, so I would like to repeat the experiment. Next time, I'm also using number of samples 3, right? And then I add them together. Is that okay? I don't know, sometimes we have to do so. Because these are all independent events, right? Each tank is independent. But then you come back to the reality that, okay, because the batch of fish are different, or different batches of fish. This fish is actually from Mong Kok, and that fish is actually from Tobo. But that could be a problem. Now you're using different fish. Even so, they're the same species. So when you do data entry, finally, outliers. Many students say, oh, this data look terrible. I just skipping abnormal data or unexpected findings. There's also a scientific fault, right? So I think I have to remind you all this. And that's why we need more number of samples in order to avoid skipping this, and then you have to do more sampling. Oh, you expect there would be a linear correlation, but at the end, may not be the case, right? In many environmental samples or clinical samples or animal tests, the error could be large. Okay, and perhaps an in vitro study would be better, but just like this example, I went out one day, that's the ocean terminal. So I went out there, I tried to collect samples in the area. All right, there's actually an outfall with water coming out. So tell me where to collect samples here. Where should I go to collect samples? According to previous research, okay, this site represents Jim Sa Zui Pier, Jim Sa Zui Matao. All right. So where should I go to collect samples, knowing that the seawater outfall is here? If I can only collect 1 sample, where should I go? Tell me. Where would you go? Whenever the supervisor tell me to do.
Place they dispose the waste water.
What do you mean? Here. Yeah, you can do so. But if you do so, then you have to specify samples were collected 1 meter away from the seawall outfall. Right? Or alternatively, actually that day we went out for some microplastic sample collection. We collect 1 and 2 and 3. Three. So a number of samples, 3 from the Tsim Sha Tsui pier, avoiding this major outfall. And then later on, after we collect sample, wow, there were a lot of bubbles. And then another example here. That's actually when I got stuck at the university station. I drove in the morning from Mount San to university. Okay, I saw this. They're trying to start a construction site to do the sewage treatment works. Okay, they want to move the sewage treatment works inside the inside this called Loi Po San. Okay, so they start to. I don't know exactly what they're doing, but I'm asked. Okay, you collect some air samples and see how that would represent whether this kind of slope projects would affect the air quality. So, what will you do? How would you go about to collect samples?
At the margins,
Hmm?
like in between the site and the tree.
In between the sites here. But there's someone is actually using this device, right?
Yeah, I just
A piling
and
device.
That standing from the standing by the trees, and then the other hand, like one side is a tree and the other side is the site.
I would, yeah, no, that's fair enough. Fair enough. Actually, the first question I asked them is, what would you like to know? I would like to know how this site would affect the residents nearby. If that's the case, I would, okay, actually there are resident blocks next door, right? Then I would make a transit from here to there and collect 3 samples, and one very close to the site, one very close to the residents, and number one in between. Alternatively, I may like to collect samples from the road and see how far it would disperse if there are air pollutants coming out from the site, right? Or maybe actually some roads down there, then I need to. What I'm trying to say is, number one, you have to identify the stakeholders who are being affected, right? Number two, you want to see, you like to collect 3 samples, right? So how or where these 3 samples will be collected. Maybe I only collect 1 sample, but then I will do it in the morning, in the afternoon, and also at night. Right? Obviously, perhaps at night, we don't see the air pollution anymore, right? Of course, best you can do is that I have 3 sites, and then I would do 3 samplings per day. Okay, then you ask for more budget from the drainage department. I need to collect at least 9 samples to represent what is happening here. All right, one more. Here, this is Dai Po. I went out to Dai Po Lim Chun River. And then according to the drainage department, drainage department again. They are telling, people are complaining this water is so dirty, and the whole channel, or canal we call it, channelization, right? They make all this river, they call it river, but actually it's cement. Very dirty. And then one of the complaints is that, because of the tidal flush, high tide, all the dirty seawater from Tolo Harbor will come in. So the day we went out, actually, the drainage department, look at this, it's so clean. So my question posed to you is, how are we going to collect samples? Considering even high tide or low tide, I'm asking for the question location-wise, where? Should we collect samples here, there, or down there? You have any idea?
I think you collect the sample in different time period.
Okay.
And when you should collect the sample when the water from the toilet well came in, and after the water when the tides go away, and then make sure this, and then collect the sample after a certain time to make sure the pollutant is not came from the total harbor.
Okay, fair enough. But you know, actually, this site is actually included in the water quality control zone. If you look at the EPD, they have the fresh water quality. This site is one of their checkpoints. But of course, it's owned by EPD, not studied by drainage department. Okay. But then if you look at the EPD website, you don't know exactly when or where, either here or there, they collect the samples, right? Of course, you may write to them and then they will say, we do not have resources to do so many sampling. So therefore, only 5 ponds are drawn from the Lam Chun River. And then from inside the Lam Chun down to the mouth of the Lam Chun River, outside is the Tolo Harbor. Without touching Tolo Harbor, but you know that the seawater may come in with tidal flush sometimes, right? And then they have a second set of data called seawater data, not included in the freshwater data. So when you read this data, you should be very careful on this. Same thing is true with the question that I posed to you last week, but no one did the discussion. That was about the tear gas problem, right? So since tear gas were dispersed in different ways, sometimes with gunpowder, sometimes it's just burning. So how are you going to go about to collect samples? Then you have to ask yourself, what kind of study would you like to do, right? Oh, I want to study how this tear gas would affect residential area. If you are going to do so, you have to collect samples from the balcony or from the window, right, of the lower floors to see whether those people have been affected. You cannot just go around and just collect some dust on the ground and okay, but if you would like to study the persistence of the chemicals, then perhaps on day 1, and then 1 week, and then 2 weeks, and then 3 weeks, and then 4 weeks later, you go to the same site. But still you have a problem of the change of the environmental conditions, right? Or after heavy rain, nothing could be found. So the other conditions, or what we call basic parameters, like if you collect water sample, the basic water parameters will be considered as well. All right. The final topic is on falsifying data. I think it is so obvious. You make up the data, or fabricating the data, or even fabricate the analysis. In the US, they have cited many different different cases, and then they present the case, how to avoid it. But if you read those cases, you will see that sometimes there's the problem of the integrity of the researchers. So in the coming questions that I pose to you, I would ask you how to impose certain guidelines to avoid such things to happen. And there are also what we call the covering up or misrepresentation. It's okay, you collect a few samples, but then you pretend that you have done all these samples over the years. And then you have to look at this. I'm not trying to go into the details of these different cases, because on each website they would explain all these different cases. So again, before starting your research, I think it's very important, after you have heard what I've been talking to you, design your experiments with good justifications. So you have to ask the question, why are you going to do this experiment? And for what purpose? Okay? After you have learned that, then perhaps you may apply for funding support. And then you should obtain relevant safety and ethical approvals, be that an experiments to be conducted inside a lab, or be that a field study, That if it's field study involving in samples relating to like air samples, relating to EPD or relating to AFCD or relating to different government departments, you should also obtain their approvals, right? And then there are 3 phases. The first was the preparation. And then 2 is during the research, you have to manage the data and log. Like for example, I need to go to collect sample from the drainage department of the sewage. Then you have to explain to them what is the best time to collect the sample, and when and where. There are different sites in there. Some samples are after treatment, but not discharged. Some samples are collected from the discharge site, exactly the location to be with the sample to be discharged and so on. And then at what time? So you need to prepare a worksheet, managing data and log, with a worksheet that details all the information. And then you have to explain to them what kind of method are you going to use, what device are you going to use, right? So like for example, the case of studying cyanide, that could be a problem of tear gas. Then you have to explain to them what kind of devices that you want to go for to do the detection. Same thing is true if you study noise pollution. EPD only recommend 2 or 3 certified machines. Then you have to use those exact machine, and or the method, is it the standard method? Like for example, those are standard method for measurement of noise pollution. Then you have to obtain from the machine the copyright of the use of that program to collect data, and that could be costly. Okay, and then we mentioned the use of personal data and information if you are involving in clinical samples. And then 4th, correct use of research funding. I've seen cases that some researchers, they obtain a huge amount of research fund, but they did not know how to spend them. Okay, and then they, every day our colleagues are debating with the account office how to use this money. There are different research grants, they have different requirements. Okay, usually spending money could be a problem, very difficult problem. And some researchers end up saving a lot of money, not intentionally saving the money, but the account office would not allow them to do, because according to that research grant, that sort of money could only be used for like consumables, for equipment, for personnel. Even for personnel, that could be only applied to short-term RA or full-time staff. They have all those requirements, and that these are very important. So I think the responsibility of research staff is also very important. We usually need to hire a project manager to go over these complicated steps. Otherwise, oh, you are my student, you come over. I just pay you from my pocket, and then you do whatever I tell you to do. Right? That could be a problem. And you have to be very careful. Of course, if there's not from a research funding, that's fine, but if from research funding, all the expenditures have guidelines to spend, and then you need a manager to help you do all this job, okay? And then you have to specify the collaboration, supervision, and division of labor. Okay, I have 5 students, you guys do those RNA study, and you guys do those experiments. And they will do the data analysis, because that involves bioinformatic analysis. There are very clear collaborations or, elaboration of work. And then finally, after the research, after you have done everything, how to publish the data, how to do data analysis. We briefly mentioned before, you write up, and then the authorship responsibility. Some journal even put down who designed the experiment, who performed the job, who analyzed the data. Okay, they even specify the percentage of the contribution. Oh, I contribute 10% of the time because I only designed the experiment, I only wrote the research proposal. I'm not the one that is involved in doing the job. Whoever is doing the job, they must declare, okay, I'm doing the job, I collect the data. But after data collection, or after sample collection, I'm not the one analyzing the data. It's someone else. Then you have to specify, then make acknowledgement of funding source. Okay. Well, obviously, it's related to the what we call the conduct issue, right? If it's funded by a drug company, then you have to declare conflict of interest. Yeah, number 4. And then you have to make data available. Now, that's the key issue why we have all these guidelines. All these guidelines must be observed because there's a funding issue here. One of the videos that I post onto the Blackboard is from the US. There were laws that Obama, at the time of Obama, they put down all those criteria. Why you have to publish this data? Because this is from public money. Why can we read all the information from NCBI, National Center of Biotech Information? Because it's 100% funded by US government. And then US government say, okay, we are for the people and by the people, so everything is open. Anyone could click on it to read it. But not every single organization is doing the same. Sometimes you need to pay to see the data. Okay? So making data available or open access, and then having the research community as advisor or reviewer to provide fair comments. If someone else doing similar job, you're supposed to give comment or help them. Okay, you cannot say, oh, I am the only one doing this job, how dare you would like to repeat my experiment. You cannot say so, right? You don't have the right to do so. You are even supposed to help others to do so. Okay, I'm very good at it, I clone something. Look at the synthetic biology community, IGM. They spread out all the DNAs. Sometimes you test it, oh, the submission is actually really bad. I'm not so sure whether they have, what kind of DNA they have submitted. But still, you have to open to everyone. All right, there's a company, not really a company, a community called AdGene. They collect a lot of DNA materials or plasmid material just to make the research community available in using all these materials. That it's also very important. Okay, so with a research code of conduct, you cannot say that I'm the only one do the experiments, I don't want others to use it. This is unethical as well. Okay, so I think I have explained all the cases here. We still have 15 minutes and then we may go over the discussion. And again, 1, 2, 3, 4, 5 is easy to find. We have for these different questions. Right? The first one, this scientist is commenting or criticizing government policy, yet he's doing a research for the government on a similar topic. So it is ethical. Number 2, this is a new concept called research impact. So we want to do research, we want to make the world better. We don't just want to publish the data and then go away, right? So could you name 1 or 2 research that you think would have significant what we call impact? That could be environmental, could be chemical, that would solve a real world problem, okay? Number 3, how to reduce those data. Many published data, we say, oh, this is not reproducible. Those guys just publish the data, and then other scientists try to repeat the experiments but could not get all this data. Could you write some guideline on standardizations or method, making sure that all the published data are reproducible? So like in my case, in my lab, I usually would ask my summer student to repeat somebody else's work done previously. To see whether different hands get the same results. 4, interlab verification. And sorry about that, I also name IGM again. One of the requirements of the competition is that they will ask different teams to test different biobricks or test the same biobrick. Now, my question posed to you is how to avoid bias. Oh, I would expect this is the IPPG inducible. So obviously this will give results, right? So what's the guideline? For example, give you a hint. You just don't tell them exactly what promoter is it. You have 5 promoter A, B, C, D, E, and then you have 4 chemicals. And then in different teams you change the name, right? That would be a good way to do inter-lab verification. By the same principle, number 5, how to avoid making up or fabricating data. You know what? In all clinical lab and all environmental samples, they only have barcode. They would not tell you exactly, oh, that's the control, and then you do it. Well, how come this data is so high? It doesn't look like a blank control. You're not supposed to know. What kind of samples are they? You are supposed to handle those samples with barcode if you are in chemical testing lab. Day 1 or clinical testing lab. Day 1 you have all the samples and then you use the scanner. And then you put all these samples in the line and then to go through the machine. And then data come back again. Even if you produce the data, you don't know exactly what these samples were when you put into the machine. Right? These are one of the, or some of the guiding principles. So could you think of others? And same thing is true. Send me email. So, who would like to start with number 1? You two? You two want to join them? No? Or this? Yeah? 4 together. And then the second question. 1, 2, 3, 4, 5 in the middle. 3. 3 and 4 could be together, or just 3. You have 4 on top and then 3 at the back. So now we only have 4, right? Okay, we can just select which one you want. Or you may combine 4 and 5 together. How's that? Number 1. Hey, only 10 minutes left. I talked too long. Okay, so that is the end of the lecture, and we'll have to go over the discussion. Now send to me email, and then I will compile